First of all, thank you very much for everyone who is now watching and listening to us. And I hope that we can give some information, interesting information to everyone and create some collaborations for the future. Florabio is a biotech R&D company registered in 2016. Total investment value is over 3 million euros. We have 13 employees and we have a 350 square meter of cell culture area. The most important information about Florabio is we are settled on the paradise side of Turkey. This is a beach only 3 minutes away from the office, so you can easily imagine that our employees are enjoying the life, not only developing good products, but also having a good life quality. I am the CEO of the company, and I am the CEO of the company since 2 years, and I have been working for sales manager of Sartorius Stedim Biotech Turkish office for 16 years before. My partner, Dr. Aziz Kayli, is the scientific brain of the company, and he is well known as a cell culture guy in the world. In fact, the company he established in 2005 called Celka was bought by Sartorius in 2015 and still known to be one of the best cell line development companies in the world. He is also known to be one of the top cell culture media development guy in the world, and the product he developed in 2008 called ActiPro is still one of the most preferred and one of the most sold media under a large supplier company in the world. We have another colleague in our team who's responsible on DSP. He is Dr. Joachim Walter, who has experience for 18 years in Boehringer Ingelheim. He was heading the process development team, and he had different various roles in different big companies, like he worked in GE Life Sciences before for 4 years, and he was also the senior vice president of InnoBio in Malaysia. Now he's giving consultancy to several consultancy services to Chinese companies in China. So Florabio is focused mainly on USP and lately on purification. We have already established our platform technology for the complete upstream process, and Dr. Walter is now developing our technologies for the purification side. Purification technologies and platform are still under development, but for any kind of upstream process development services and product developments, we are working very intensively since last 3 years. Instead of going into different fields, I would like to focus on the main business model of Florabio, which is media development and which is host cell line development. And I would like to start with a cell line with some products which are really hot nowadays in the market, not only used for COVID-19 vaccines, but used for many other gene therapy, cell therapy, and vaccines. The first product we have is Florabio HEC293 cell line. This cell line is commercially owned by Florabio. We have the complete commercial license and ownership of the cell line. We have developed this cell line to serum-free and suspension conditions. The chart here is only to show you how stable the cell line is. We could run a stock culture for 270 days, and during this 270 days, we do not see any fluctuation on viability and on viable cell concentration. Of course, we don't use this cell line after 270 days, but that's how stable the cell line is. It's currently being used in 2 different COVID-19 vaccines, which are in phase 1 and phase 2, and we are aiming to out-license the cell line to 6 new companies in 2021. Of course, this cell line came out to be a side product when we were trying to develop cell culture media. As I told before, our main intention and our main business model is to become one of the best media development company in the world. And the product that we were working on HEC293 cells was in fact the production media of HEC293 cells, and we call it ORCID. Since we are a very small company and since we are doing only R&D, we have used all our chances to get all the benchmark products from the market, which are quite well known and quite well used. And as you can see, we can outperform all other commercial products out in the market minimum to 2 to 3 times. For the first time in the market, we developed a feed media for HEC293 cells. And this feed media is mainly intended to be used for virus production, adenovirus production or viral vector production. And as you can see, together with the feed, we can take the performance, we can bring the performance of our production media up to a level which is not yet met in the market. So we can achieve up to 30 million cells with suspension HEC293 cells, which is 3 times, 4 times more than what the market average is using at the moment. We have another competitor who is also working on feed media, where they are also quite good and quite successful. I just wanted to show you the results. When we were working on HEC293 production media, we had continuous requests coming from the market if we can also develop a transient transfection platform. In fact, we were asked to develop a transient transfection media, but then after giving 3 years of R&D work, we have developed a transient transfection system, which we call it IV. And with this IV transient transfection platform, we have the cell line, which belongs to us. We have the stock culture and the cell bank, which is RCB level, and we are currently working on characterization and master cell bank, working cell bank development on this HEC293 cells of Florabio. And we have the culture media for transfection. We have a transfection standard operating procedures, SOPs. This means besides the chemical which is used for transfection, we could develop or we could bring a complete platform for quick and fast and robust protein or viral vector or adeno-associated virus production in the market. Since we are only a cell culture laboratory, we could not work with viruses. That's why we have taken green fluorescent protein plasmids to test our transfection efficacy. And according to the test that we have done with all our competitors, we have seen that we can outperform them with our cell line and with our platform. At the moment, our media and our cell line is getting evaluated at 3 different locations. 2 of them are in Europe, 1 of them is Canada. And at these locations, we are already getting some positive results that we can achieve the similar results with viruses, especially on adeno-associated virus and lentivirus. But to give you the impression how much we can achieve or how good the media and the platform is, I can easily say, or you can easily see that with the green fluorescent proteins that we are working on, we can see the green fluorescent proteins shining with our blank eyes, with our with the eye vision. As I told before, we are a media development and media supply company, and since the beginning of COVID-19, we also fasten up our development projects, our R&D projects on the COVID-19 vaccine cell lines. And one of them was Vero, which was going on. The R&D was going on for 3 years continuously. And we have also managed to have a supplier, we have managed to get a partner in the market who could supply us their suspension Vero cell lines. Vero cell lines are commonly used for inactivated viral vaccines, and nowadays we all know that Vero cell lines are also used to produce inactivated vaccine for COVID-19, but suspension Vero cell lines are quite unique. You don't see that in the market. There are only 1 or 2 companies who have developed it, and our partner in Austria is one of the most sophisticated company who has already experienced production of influenza and SARS-CoV vaccine in their adherent vero cell lines. This is a co-development what we are doing, because when we are developing the media, they're also testing the virus infectivity and also virus productivity. According to our results, we could take the suspension vero cell lines up to 2 million viable cell concentration, which is the only one, or which is very unique in the market. There is no suspension culture media for vero cell lines at the moment commercially available in the market. That's the first one from Florabio. And you can see that we can achieve up to 2 million cells. And when we compare different serum-free media in the market to our serum-free media, we can see that we can achieve at least the same performance and even a little bit better. We should not say a little bit better, but we can say we are at the same level currently, and the R&D process is still going on. We also developed, at the same time, we also focused on developing another cell culture media, serum-free cell culture media for adherent vero cell lines, which is the main cell line to produce at the moment in the market for COVID-19 and many other vaccines, viral vaccines. We have taken one product out there in the market as a benchmark, and according to our test results, we are at the same level. Together with the vaccine media development, we of course have developed and worked on CHO platforms and media platforms on different CHO cell lines. The main CHO media or cell line which is used commonly in the market is CHO DG44, and our media development performance is already shown in the figure. The black figure in this chart is the most commonly used and the most sold media in the market, which was developed by Dr. Kiley in 2008. That's still one of the best one as seen in the chart. We can outperform this performance by 40-50%. At least we can tell that we are at the same level of performance. These tests are being performed by a clone which is already producing an Fc fusion protein about 2 grams per liter. Of course, that process or that kind of processes has never done in batch mode, so we have to develop a feed on it. And with our feed, we can still have, or we can again have the same performance as our benchmark, as the media which was developed by Dr. Kiley. Here in this chart, I would like to emphasize, I would like to give one bulletin point, which is very important for us and which is a service that we give. As you can see, with a clone in our hands, which is already producing an Fc fusion protein, we could test 2 medias in parallel. And according to the test results, we can say that we are at the same level. But when we get a clone, which is already producing a biosimilar product, our expertise is to touch the media formulation and to optimize the media formulation so that, as you can see on the blue line with triangles on it, we can optimize the production yield from a process. Here you can see that with the same product, we can take this performance with our competitor product, with the X product that Dr. Kiley developed, almost 40-50% higher. That's where we are really strong on, and we can easily say that we are one of the top 3 companies in the world who can manage it. Since we were working on the virus side and vaccine side, we have been also requested to develop a media for MRC5 cell lines, which are commonly used for very big blockbuster vaccines like rabies, rubella, varicella, chickenpox. And the MRC5 cell line that market uses are always fibroblast and adherent cell lines. What we have done is we have spent 1.5 years and we have created another version of MRC5 which is adapted to suspension culture conditions. We are currently testing our suspension MRC5 cell line for production of rabies and influenza viruses. and the test results will be shared very soon. And the results, the primary results, are quite promising. According to according to the information we received from our partners, they can see that they are producing almost 90% as much as adherent MRC5 cell line in their hands. The results will be shared soon. If anyone is interested, I can also share the results with you. The interesting part with this MRC5 cell line is the cell line is growing like a bacteria. The average split ratio is 1 to 33. Once the virus results are there, we will immediately share with the public. And the media which we developed for MRC5 cell line, suspension MRC5 cell line, is called MIMOSA. And this is, again, a unique product which is only existing in Florabio portfolio. So we have made our research in the market, and we have checked if anyone is using any serum-free media, especially in China, in India, and also some of the South Asian markets. And we have seen that mainly these classical medias are used together with serum. And that's why we have tested MEM alpha, DMEM, Glasgow MEM, and DMEM with high glucose, low glucose, together with media, together with serum, and without serum. And we have decided to take at least one vaccine media, which is existing and which is one of the most common medias in the market, from our competitor, with serum or without serum. As you can see, we can outperform it. The interesting fact here is, on our own MRC5 suspension cell line, our serum-free media is working better than if we add 5% serum. In this case, we can also claim that our platform, media platform, development platform, is so successful that we can detect and provide enough amount of amino acids where they don't need high amount of amino acid and protein cocktail anymore. Since my partner is working on biosimilars and biotherapeutics for the last 30 years, his know-how on process is not on batch, but mainly on fat batch. So we always try to increase productivity by also developing a further step on the process development. That's why we have developed the world's first MRC5 cell line feed media. And with our feed media, we can take the performance or viable cell concentration performance from 10 million up to 20 million cells per ml. As I have stated, the virus results will come very soon, and if we are going to present them, we will present it as a package where cell line, media system, and the process will come and will be provided to customers, where they can benefit and reduce the investment costs up to 90%. The last product I will show is the BHK21 cell line from Florabio, which is again owned by Florabio. All the cell lines I shown are commercially owned by us. We have all the commercial rights, and we are out licensing them all around the world. The BHK21 cell line we developed is, again, suspension condition adapted, serum-free condition adapted. The average split ratio is 1 to 43, and we have a very robust viability and very sustainable viable cell concentration. The average viable cell concentration for BHK21 cells, especially for production of FMD viruses, is normally 2 to 5 million. Now, in our stock culture, we can achieve 12 million easily. And you will see that our media is achieving 14 to 16 million cells per ml on a batch. This product is already licensed out to one of the global suppliers in the world. It's sold in the market with the brand name of one of the big companies, and that information I cannot share here. And this is the comparison sheet that you can see easily, that we are outperforming them tremendously. Again, my partner decided and he wanted to bring another aspect to virus production with BHK21. Especially, we were aiming for FMD vaccine manufacturers where they have to make big investments to produce certain amount of viruses. And we were aiming to bring this feed media, which is unique in the market, the only one in the market, so that the investment cost can go down drastically. Here you will see that we can bring the performance of our batch medium, production medium, almost 40-50% higher to 22 million cells per ml. To sum it up, all our Florabio medias are animal component free, serum free, protein, peptone, peptide, lipid, growth factor free. In very simple words, our products are 100% consist of 100% single chemicals. We don't use anything from natural source driven. All the products and the proteins, even if we are using proteins for maybe for stock culture media, we are using them from a recombinant sources, which is sustainable and which is also regulation accepted. What we do in Florabio is we do cell line development. We do media development for customer specific cell line. And this is where we are very strong and probably one of the top 5 companies in the world. As I have mentioned before, any customer, anyone, especially now that I am speaking to Korean colleagues, anyone who is interested to see if they can achieve higher process yields with their existing clone in their hands, we have the complete platform ready to provide you results within 2 to 3 months. Maximum in 6 months, you will have your clone back with a product code where you can achieve up to 200% process yield optimization. We also offer process and titer optimization, not only with media development, but also with process simplification. We do upstream and downstream process development. We do vaccine process development. And one of the R&D project that we are working and we are also giving service for is glycoengineering, which is improving protein glycolization via media. At the moment, we have a European Union grant project going on where we are developing world's first media, which is going to optimize protein glycolization for any clone. That will come very soon. The last service we give is training, which is given under the name Florabio Academy. We have 11 consultants, trainers. Also, some of them are very valuable professors and colleagues and experts from Korea. And we provide 22 standard trainings. And what we do is we tailor-made trainings for customers, starting from executive board members to the blue-collar technicians. Thank you very much for your time, and thank you very much for listening to me. If you have any questions, please do not hesitate to contact us, and I hope that you will have a successful, fruitful bio.